So so this just repeats what we saw before. Here's the KDEL receptor. This is from your book right here. We see that the KDEL containing proteins, like, and you know what those are, have KDEL, they're bound to the receptor. The receptor tail has KKXX. You could draw that into this diagram right here. KKXX, and that provides a binding site for the COP1 coat. So as these COP1 coats form at the cis Golgi, we're going to capture the receptors as well as the KDEL protein that are over there. So let's answer your question now. How does this work so that the KDEL containing protein, the KDEL receptor, will bind to KDEL proteins in the cis Golgi, but then will release them in the ER? Otherwise, this would never work. Okay? Do you guys get what the problem is? How does the cell do that? So it will bind to KDEL containing proteins in the cis Golgi, but release them in the ER. And it turns out that the KDEL receptor is exquisitely sensitive to pH. So there's a pH gradient as we go from the endoplasmic reticulum through the secretory pathway. And it gets, whereas the ER is relatively neutral, you get more and more acidic the further you go along the secretory pathway. So the cis Golgi is a little bit more acidic. It has a slightly lower pH than the ER. The Golgi, and then as you go through the stacks of the Golgi, you get more acidic, with the trans-Golgi network being more acidic than the cis Golgi, being more acidic than the ER, et cetera. We'll see later that the lysosome is a super acidic compartment. So the KDEL receptor is exquisitely sensitive to pH, so that it will bind to KDEL containing proteins in the cis Golgi at the slightly more acidic pH of the cis Golgi, but then it will release them in the ER, which is more neutral. So, and this occurs because its conformation, its binding site for KDEL is exquisitely sensitive to pH. It's just a couple of tenths lower pH in the cis Golgi, but that creates a conformational change where the KDEL binding pocket in the KDEL receptor will no longer bind to KDEL. And so it will release the KDEL, and then we'll go forward without binding, and we'll go back to the cis Golgi. And you can see what this slide also illustrates is that COP1 coated vesicles are bringing KDEL receptors along with and dragging along KDEL into the ER. And then the way that the empty KDEL receptors are recycled back is through COP2 coated vesicles. What this also illustrates is that so most of the KDEL receptors are in the cis Golgi, but with some frequency KDEL containing proteins, maybe they don't bind to receptors there, and so they'll go further up, and so you can find lower concentrations of KDEL containing proteins in other stacks of the Golgi. But it's very efficient so that you don't end up finding KDEL containing proteins in the extracellular surface. They're all returned back to the ER. Questions? Yeah?
So when you're saying pH gradient, that's different, lumen, like inside?
Correct.
Not the cytosol.
No, the cytosol is neutral. That's absolutely correct. So this is all neutral right here, and this weak gradient, which is only a little bit in each compartment, means that the lumen of the cis Golgi is slightly more acidic than the luminal environment of the ER, and the stacks get progressively, the inside of the stacks, the lumen of the stacks get progressively more acidic as we go along. We'll see that one compartment later, the lysosome, which is further along the secretory pathway, is super acidic. Yeah?
Is the acidity maintained by the proton pumps?
Oh, really good question. So what maintains this acidity? And yes, what maintains this acidity is proton pumps. So we have more proton pumps the further you go along, and these proton pumps are embedded in the bilayer of these compartments. Question?
Follow-up question. So if there are proton pumps in the bilayer, how is it ensured so that they don't butt off with the?
Yeah, okay. So how, if there are proton pumps in the bilayer, I love the way you're thinking, how do those escape the vesicles so they're not going to further compartments? And it is easier for a transmembrane domain protein, like a proton pump, to escape being captured into vesicles, because although we have drawn these as, you know, flat bilayers that where proteins might be evenly distributed, there are actually areas of each of the bilayer, whether we're talking about the plasma membrane or the Golgi, that are enriched or lacking particular proteins. And transmembrane proteins can be easily tethered into or away from those compartments. The soluble proteins don't have that advantage because there's no way to tether them. They're soluble and liquid and floating around in the liquid. Excellent question. Does that make sense?
Thank you.
Other questions? Okay. So that's how proteins are retrieved into the ER. So let's go to another Instapol question. So COP2 coated vesicles contain all of the following except. Okay, so we originally had just about every answer picked, but then we've converged on this answer right here. All proteins listed above are in COP2 coated vesicles. So anybody want to tell me why they picked that last answer? Go ahead.
So we know that beta receptors are within COP2 vesicles, and we know that COP2 is anterograde, so proteins will be or can be destined for the extracellular matrix. Plasma membrane receptors, we kind of alluded that to how we were creating transmembrane proteins, and Golgi enzymes. Those proteins have to be first manufactured in the ER lumen before they're transported to
Mm-hmm. Everything along the secretory pathway has to begin in the ER. So I like your approach. What you did, and this is a great approach for multiple choice exams, and 3/4 of we'll release the exams as a practice exam pretty soon for the exam that's next Tuesday. But your approach is a wonderful one to take with this. You go, instead of, and often we have distractors on here, instead of like looking and say, Golgi enzymes, okay, so we haven't talked about those, that must be it. But you went through each one and explained to yourself which ones were, rather than narrowing on. And you sort of crossed out these ones, so it had to be that one. That's a great approach for exam question, and your logic was correct too, that all of these proteins have to begin life in the endoplasmic reticulum, because everything along the secretory pathway has to begin there. So all of these proteins are going to have either an ERSS or a start transfer sequence if they're transmembrane domains, if they have transmembrane domains, to place them in the bilayer. If they're soluble, they're going to have an ERSS. Does that make sense? Question?
So the KDL receptors, they dock in the ER, like from going through the Golgi, you know what I mean? So they
Like right here?
Yeah, so they like dock in the membrane and then then they get recycled back through the system.
That's right.
They don't let the vesicle and take it back with it.
I don't know quite what you, so so when they're initially synthesized,
Yeah.
they are synthesized in the endoplasmic reticulum, and then they get up to the cis Golgi by going into COP2 coated vesicles, and then they're doing their job by having signals here that make them go into COP1 coated vesicles. Does that explain your question? I didn't quite get it about the vesicles. So there are two kinds of vesicles, right? They're going to be in COP1 coated vesicles, and they're going to be in COP2 coated vesicles. And they initially, before they grab anything, are going to be synthesized into the ER. And with all the other secretory proteins are going to be in COP2 coated vesicles. Perfect. Yeah?
The KDL receptor that goes to the ER, that comes from this Golgi and
This guy? Uh-huh.
is that recycled, or is it a new K-Del, or is it the entire curriculum just making K-Del recycled?
Okay, is this one right here recycled? And the answer is, it is recycled. So it'll be very inefficient to keep making these proteins, and so the cell conserves energy by recycling this protein continuously. And it recycles, it's able to recycle because it binds only in the cis Golgi, and then will release the protein, the K-Del proteins in the ER, and then the COP2 coated vesicles are going forward right here. And you can see these K-Del, these whip-thick proteins are meant to show the K-Del receptor, K-Del containing proteins, and they are soluble in the ER. Excellent. Yeah. Question here.
So like how the ER is K-Del, does the Golgi have anything to keep its proteins that it needs inside the Golgi that transports it back?
Does the ER have,
Does the
say that
Golgi,
question again.
so like the K-Del has, or the ER has K-Del to keep the proteins that it needs there. Does the Golgi have anything to keep its necessary
I
proteins?
like your thinking. So you're realizing this conundrum that cells have with the traffic is all going this way. And that's how the ER deals with this. How does the Golgi deal with this? And that's a little bit less understood. It's gone in detail in your book. It turns out that the Golgi has very few soluble, maybe none, soluble proteins, soluble resident proteins. So what would be some resident proteins in the Golgi?
Well, I'm just thinking some for like glycosylation.
Absolutely.
Like
So you're remembering that there's extensive modifications that go on in Golgi. So there's a bunch of enzymes there, resident Golgi enzymes. Your question is, how do those stay in the Golgi? And it turns out that very few of those resident proteins are soluble, so they're not facing what the K-Del containing protein is. But you're also thinking, well, still there's the bilayer sort of moving. How do those proteins retain? And it turns out that there are probably, it's easier to retain membrane proteins because they can be excluded from sites of exit. And this is actually a great place to end because that's where we're going to start in the next class with what happens in the Golgi. Okay.